And now it is a great pleasure to welcome our today's speaker. So Jack is a senior scientist since 2020 in the computational biology group of ETH Zurich in Basel. I always say like ETH Zurich in Basel at the DBSSE. His research interests are around cancer evolution, modeling, phylogenetic tree inference, network and probabilistic graphical models. And then he also is interested in single cell sequence analysis. But the first three you see, there is inference and models involved. So Jack has a very strong background in statistics. He's also teaching statistics, has this very cool stats clinic he's hosting. And I myself did some causality during my PhD back in the day. So I'm very excited to give you today's presentation. And well, without further ado, Jack, the floor is yours.
Yeah, thank you very much, Daniel, for this nice and detailed introduction, especially the pauses in between the research topics. Very much appreciated. I hope you can hear me okay. I'm using an external microphone, not as good as your one, Daniel, but maybe it helps with the quality of the sound. Okay. Yeah, so I say thanks very much, Daniel, for the chance to speak here today. I wanted to talk about using these tools for computational reproducibility. As you can see in bold, I want to focus on using it for causal discovery. So before I get into the tools themselves, which probably are more familiar to people listening to this series, I wanted to dive a bit into what we mean by causal discovery, what models we use, how we can learn these models, and then how we use these tools for benchmarking. So if you like to set the scene, a causal diagram is, in the old days, was known as a Bayesian network. So these are directed graphs. So in the sense of graphical models, we have a graphical structure. The nodes are random variables, or if we look at data, these are the data we observe, we measure. So each node would be one measurement. Say if we look at gene expression, each node would be one gene, and the value on that node is the measured expression for that observation. And then the nice thing about these networks is that they encode dependencies between the variables. So conditional dependencies between the variables, and likewise, they encode independencies. So in particular, each node is independent of its non-descendants given its parents. And one key property for this to work is that the DAGs, or the graphs, are acyclic. So if I start from one node, I follow the edges along the directed arrows, I can never come back to where I started. So with this property that I can never return where I started, you can factorize large high-dimensional joint distributions into this graphical representation. So a key thing we use them for is reducing high-dimensional data down to a neat, compact representation that encodes all the dependencies that should be in this high-dimensional data. Another thing you can use them for is for causal inference. So you have these edges now have a direction which has a meaning that one node is a direct cause of its descendant. Now for causal modeling, there's more tricky things. So before we get there, maybe I'll talk about one of the challenges of learning these things. So if I draw these networks, they're quite nice. They have a nice property. You can generate them recursively. So Robinson did this in the 70s in a series of papers in combinatorics journals. And the number grows very quickly. Because you can generate them recursively, you can actually sample them uniformly very, very cheaply. You can generate them at random nicely. But we don't want to work with no data. No, we don't want uniform graphs. We want to learn them from data. And then when we learn it from data, the difficult things are, well, one, the number grows massively. So this number 10 to the 80 is a very large number, even with very small number of nodes. And this acyclicity property is not local. It depends on the entire structure of the graph. And this means, well, actually, in general, if you want to learn it from data, how some data want to learn it? Also, I got confused. It's two things. One, it's a hard problem, but before we even get there, I said we use these things for causal estimation. If we want to use it for causality, we have to make some very strong assumptions. So basically, these assumptions I've written down here. Out in text, if you like reading, you can read along. But the basic assumption is that you're modeling a system where there is a causal underlying mechanism. So first assume that one exists, otherwise you can't hope to learn it. The main thing here is about faithfulness, that your representation encodes all the distribution, all the independencies in the data can be represented in the graph and no other ones. No, this is quite a strong one. And the main one, the one we should always worry about in real data, is this causal sufficiency. That in our network, we have every measured variable that has any effect on the other ones. No? So there's nothing hidden. There's no unmeasured confounders. And of course, in real data, this is the one that's most unlikely to hold. No? There's always something you've not thought of or not accounted for. And this can completely mess up all your causal reasoning. I see Daniel was enjoying this. So this prompted, although they're kind of popular for causal inference, now if I know the structure, I know what depends on what, I can use it to put my assumptions down in a graphical form and use it for causal inference. If I want to learn it from data, then I need all these assumptions to have any chance in doing so. And even if I have these assumptions, and even with perfect observational data, there's another issue, is that some graphs we can't learn, some directions we can't learn anyway. No? So if we go back to the original representation of a factorization of a large distribution, there may be more than one way of doing the same factorization. And in some of those things, errors may go in different directions, and this can never be resolved from observational data. Then you need interventional data. So these are kind of the assumptions we kind of sweep under the carpet a bit, if you like, that, you know, all these things happen. If we assume this, we believe this, we can then try and learn this from data. And then we come to the actual computational task, and we find out it's not an easy task. It's a hard task in general. So given any data set, to learn the best network or even good ones is not easy or not possible, typically in polynomial time. So because it's a hard task and it's useful also for causal inference now, there's been a lot of research in trying to find good ways of doing this in reasonable time. And there's different strands of inference that exist. So one is to use this independency representation to try and learn from the data independence relationships. So test for dependencies. If I find it's not dependent, then I can remove edges, basically reverse engineer all the edges from testing many sets of nodes. So if I can find a subset where it's independent, I can remove some edges. One difficulty here is I may need to do many tests to check all the edges that could exist, and this can be prone to errors. If I do many tests, I find false positives and false negatives. Another approach is to define a score. So for each network, each graph, I can say, how well does it explain the data I do observe? So in a likelihood setting, what's the marginal or the maximized likelihood of my data given my graph? And this is easier to define if I put in a model for my data. So one nice thing in these Bayesian networks is they allow factorization of each child given its parents. So I don't only need to define a model from each child given its parents, so a local distribution of those set of variables. And then if I can do this, I can maximize and penalize, or I can marginalize if I want to be more Bayesian, and I can look at the posterior graphs given the data. This is your scoring function, a measure of how good a fit my graph gives to the data. And then this is then really an object in classical combinatorial optimization. So in the computer science literature, there's been many approaches to try and do this hard task as well as you can, though, from basic gradient hill climbing type searches to dynamic programming and ILP things, which can be exact as long as they complete. And in general, because it's hard, then these things stop working at some point. What's nice is also to combine these two ways of looking at things. So to take constraint-based methods to reduce my search space. So instead of having such a massive number of graphs to look through, to make it more manageable, and then inside that to search in a smaller space. So this constraint-based testing can be quite quick and then have a more accurate search inside. One thing that became very popular recently was embedding this problem in continuous space. There was a NeurIPS paper, not this one, an earlier one, which proposed this, and it got very popular in the machine learning literature until these people realized that actually they were not comparing themselves fairly when they generated the data. They were using this information implicitly without knowing when they learned it. So they looked like they were doing well, but when you compared it more fairly, they actually performed quite poorly. So there's a big hype, it's still very popular, and there's a whole little chain of machine learning papers that follow on from this and keep going along this direction. So it's a nice idea, but of course you can never get past this hardness. If I put it in a continuous space, I just have a very complex, non-convex landscape to optimize, and I'm stuck in that case. what we try to advocate, we come from a more Bayesian setting, is not just to find one graph which maximizes the score, but to sort of understand the uncertainty in this space, so to sample graphs and get an idea of the distribution of graphs given the data. So despite it being a hard problem, and in general it's very interesting, there's a lot of work in finding good or fast methods for doing this. I wanted to highlight, before I get into this and the computational reproducibility, some applications that we've been using of these networks. So the first one is we want to understand complex high-dimensional data. So here on the top right, we have data from the TCGA on mutational profiles of each column. Here is a patient. The colored bars are different cancer types. So we looked at pan-cancer analysis. And you can see this is the raw data. The little green dots are mutation present, white is absent. And you can sort of say, I want to represent this big binary matrix as a network. You may ask, why would I want to do that? No, I have a nice binary matrix. And maybe you can see here that these dots are not independent, but there's some dependency between it, and then we draw it as edges in this graph. Now you can imagine if I learn a network for each of these, I have many networks, all with hundreds of nodes, and I want to somehow visualize this mess. So here, you can think of this picture just an extract of all this information. We focus on the most important edges and genes for each cancer type. And it gives you a sort of overview of the importance of the genes in terms of their dependency structure with other genes. So these mutations do not occur independently, but if I know for colorectal cancer this mutations here, it tells me information about this other mutation. It's more or less likely this one is also mutated. And also higher order dependencies can be encoded in this graph structure. And so you get an idea across many cancers of which genes are important. Also the size represents the frequency, which interaction is important. Some of them more localized to a particular cancer type, and then some play a role across many cancer types we draw inside the central hub. This is kind of nice maybe to draw a pretty picture. It's not very useful for doing much else apart from looking at different genes. And one nice thing you can do is, because these networks are, once I learn the structure and I learn the parameters, not just a graph, but a generative model for more data of the same type, I can remove, if you like, these labels on the top, and I can use it to recluster these profiles, try and find groups of similar profiles that may be similar in terms of mutations, and hopefully similar in terms of outcome. So for this data, we haven't got very much treatment data, but we have survival outcome as a proxy. Before I go into that, what we realize is we can abuse the causal representation to help us do this clustering more cleverly. So there's some variables that affect mutations, like the stage of the cancer. Later stages have more mutations. Older patients have more mutations. The tissue type also affects which genes are likely to be mutated and selected for in cancer. And if we account for this information in the network setting, I can use it as a not hidden confounder, but an observed confounder. I can use it to help me learn the structure of the mutations better because they depend on these factors. But I can also use it in the clustering to not include it as another variable, not just concatenate my data, but I can use it to adjust. So I find clusters which are kind of conditioned on these things and not driven by these things. So we kind of leverage the directionality of these networks in a computational way to help us do this clustering. And in terms of predicting survival, which is here the only real proxy we have on this large scale data, we find it actually does help. If we just look at the mutations, this can predict survival quite well. We tested in the original paper against many other methods. This was actually quite a nice method in terms of survival. If we use this clinical information in a graphical and directed sense, it actually helps us cluster even better and predict survival for these patients more nicely. And the other direction we've gone in, if we really go into the causal interpretation, I can use this not just to learn a network. So here I have an example from psychology where we have observational data from UK population-wide surveys. They're asking about psychological symptoms and other things. So you can learn a network across this data, it's cross-sectional data, with all the assumptions we had on the previous slide. And if you believe them, then this would be a network representing the dependencies between these variables. In this case, we looked at the effect of childhood bullying, which is antecedent in time, at least, on current symptoms measured in the population. And already here you see in these light blue boxes, there's certain things we can't learn the directionality. But either direction will be equally likely, or not even identifiable from observational data. But you can still see, despite this, there are some cascades of information that go down the order in the graph. But rather than just learning a network, we can go one step further and say, okay, I can go in here using Perl do calculus, say, if I were to intervene on a variable, given its model and the structure and the learned parameters, I can then estimate what the effect would be of that intervention. This is what we see on the right-hand side. Now, if I were to intervene on the row, change it from 0 to 1 in this binary data, what would be the effect on the response on the column? And you can see here, because it's in a Bayesian setting, we sampled graphs. From a graph, we learn these or sample these effects. So we get here, by marginalizing out, ignoring the graph, we get the distribution of intervention effects or putative intervention effects. And this is kind of quite nice in the sense that we have this observational data, we run this procedure, computation is quite cheap, and we get an idea of what might happen if we were to intervene under strong assumptions. So we may not expect it to be what actually does happen if I were to go and intervene, but if I had to prioritize, which trials I'm going to run on patients or people, which are quite expensive, I might think, okay, these ones upstream may be more likely candidates to have an effect, and where they might have an effect than the things which here look like they're downstream. So we can use this as a way to kind of prioritize actual clinical trials to validate these predictions. So here I just wanted to give you an overview of where we're using these networks. But of course, along with the assumptions, we want to make sure the networks we learn are good networks. They represent what's actually in the data. So to do this, we want some idea, if I generate data, if I know the true network, how close can I get to the network? Now, this is a challenging problem in many ways. One is there are many algorithms that exist. It's been a popular field in computer science and machine learning and statistics for quite some time. Many algorithms exist in a domain. And if I want to compare them, it can be pretty challenging. So not all these algorithms in the same language. Different people, especially different fields, like different languages. They often have very different input and output formats. And if I want to compare large scale, many algorithms, I need access to high performance computing, parallel computing to make it worthwhile. And then if I run all these things on a large scale, I need to structure my results, put them together so I can then actually compare it in a neat way. And of course, if I want to compare, I need to choose a metric. And even if I can do all these things, if I want to actually do it, it's going to take me a massive amount of time. So we came across this problem. So actually, when we were trying to publish our method for sampling, it's a problem we faced. We did, like most people, we compared to some selected competitors. We run a small scale simulation study, and everybody does their own simulation study, and then it's very hard to really compare across. So I took this picture this morning. It's a bit like the Wild West, where everybody's doing their own thing, and there's no real structured comparison. And of course, if I just focus on one area, I can look better, but maybe I'm misleading myself and others. So this is the stage we were at a few years ago. How can we do this in a more sensible, systematic way? And this kind of made me think of when I first joined the bioinformatics field. So about 10 years ago, I came to this field. And in those days, maybe people here remember what it used to be like in the good old days or the bad old days. If you wanted to do any bioinformatics analysis, you typically have a large string of interdependent steps you need to do. So you start with your sequencing files, quality control, aligning, go back through sorting, organizing, removing unwanted things, and then in the end, maybe you call the variance, and that's what you report in the end. And keeping track of all these steps used to be done with these very complicated bash scripts. Everybody had their own personal version, and each lab had their own bash script that was passed on from one person to the next. And in terms of reproducing other people's work, this was always a nightmare. One, getting the data, getting the scripts, getting the right versions working was, I think, unlikely to work very well in practice. And what happened in the bioinformatics field was the game changing of these workflow managers and containers. So I can make all my steps reproducible in a container, and I can keep track of every single step with the workflow managers. So this is what kind of inspired us to use these things and bring it to the world of causal discovery. And the main thing, of course, that we use for this is the SnakeMake workflow. So I guess this was the last seminar in this series, was by Johannes Costa about SnakeMake, and it really has completely revolutionized bioinformatics. Instead of random bash scripts, it now is the de facto way of doing analysis in a reproducible, sensible way if I want to share it and make it useful. And Johannes, I guess, because other people have used this. I mean, here you see the citations. It started off, 10 years ago, grew very quickly. Now there's other papers, so I guess it's tailing off because they cite other papers, but still it gets one citation a day. GitHub is very, very popular. And the neat thing is you just need to say, in my massive workflow, each step I can define what's the input, what's the output. So I guess you heard last time from Johannes now, I have a JSON interface. I just define these things. And then a really neat thing is Snakemake says, okay, I take all these dependencies, I work out how they're related to each other, and then I build you a DAG of the relationships. And then given that, you can submit the jobs, wait for the outputs to start the next one, and collates everything for you. So this makes life so much easier now for bioinformatics. And we wanted to bring this to the world of causal discovery. So what we did, well, when Felix, I'm not sure he's here, maybe he's in the call, he joined Juice's group at University of Basel. And we were like, okay, can we take these tools from bioinformatics and bring it across to causal discovery? And so we developed this kind of functionality. We call it bench press. And we exactly use the Snakemake workflow as the heart. We use everything that's been developed by other people to make it easy for us. And it means we can do the internal bits in any language using these Docker containers. So we can take all these different algorithms in different languages, put them together. And then the nice thing is you have to just define your modules now. So I'll go into this in a bit more detail later. We need a module for how do I generate graphs, generate data and parameters, how do I learn them, and how do I benchmark, how do I compare different algorithms. But once I define that, then everything's done in its workflow manager, can be a parallel. Yeah, you can reproduce it and share it. Now what I need to do is share the file that sets it up and the whole thing, the container, and the file is enough to reproduce everything. And the neat thing is it's done with these JSON files. So that's everything I need to look at is in these simple files. Yeah? All the details I have for my benchmarking stored in one of these files. And this and the results can be easily shared. And then the nice thing is because it's modular, now you can easily add new modules, new functionality, and I get to that at the very end. So to give you an idea of what's in BenchPress, what we need for causal discovery, here is a kind of overview of the different modules we have. The top ones in pink are about generating the data. So the main thing is how do I generate a graph? And there's different modules that do this, different ways of generating random graphs based on different packages. If I want to benchmark for many seeds, now I need a range of seeds. I need to generate for each graph some parameters. And then from that, I can generate data, different sizes. And then I can also look at my original data and I can look at how it's distributed. I can look at the true graphs. And the thing we maybe care about more is given this data, how well can I reconstruct it with these algorithms? I'd say there's many of these things, and they're all built into the system. And then depending on what my algorithm does, I can look at, the output graph. I can look at how expensive the algorithm was. And if we go for the more Bayesian setting, look how close am I to convergence with these sort of MCMC type plots. And then, the final thing in purple is how do I benchmark it? What do I compare against what? So all these modules exist. I'm going to go into a bit more detail of some of them in the next slides. And the thing I wanted to really focus on is, as in the title, how do I use this for benchmarking? How can I test, is my algorithm a good one? How can I pick a good algorithm for my downstream analysis? So ones we use in oncology or computational oncology or like in computational statistics. So we kind of defined, if you like, 5 main scenarios. So the first one is just a pure data analysis thing. I have some data that's come from some field of study, some domain. I have the data, I want to see, I can analyze it with any one algorithm quite easily, but maybe I want to see how robust is my analysis to the choice of algorithm. So in this BenchPress world, I can take the same data, I can run it through every algorithm that exists more or less. And I can see how similar is it across all different algorithms, what's robust, and what depends on my choice of algorithm. What's a bit less likely is the second scenario where the data is given and people think they know the true graph. So you have sometimes these kind of gold standard networks in the literature and the data, and then people say, oh, I should try to recreate this one graph. People who are more used to these things really know that the ground truth is often very far from the ground truth, but at least you get an idea of how far away your learning can be. Whereas what we particularly care about maybe for benchmarking is this more random setting. I generate networks, generate data, and I can see how well can I recover what I started with. And I can see how it depends on my graph structure, my graph parameters, my local parameters, and the data, of course. Now, here well, we particularly like the completely random setting. Well, I pick a random graph, maybe depending on some hyperparameters, generate data and check how well can I recover this. The machine learning community tends to like their favorite set of known graphs in the literature. So there's some legendary set of given graphs that exist that they all like to generate new data from and try to recover this set as if. And the nice thing is, or maybe the weird thing is, if you go back in the history of these graphs, often they were somebody had some data, they learned a graph once with one method, it got published, and then it got taken away, and it's become, this legend is lost to the mist of times where it came from, and now it's a standard graph in the literature. So you can do this, and the machine learners love to do this, but we prefer making everything random and seeing how it depends, say, on my density of the graph. If I change the type of graph network, how does it affect my results? And then once I generate data, maybe, sorry, go back here. The core of this is really comparing all these different algorithms. And to give you an idea of the scope of these things, here I'm highlighting just a few, but currently we have about 50 algorithms for learning directed graphs, so these Bayesian networks, and then another 10 or more for undirected networks, so a different model class. And here I just want to go through a small subset to give you an idea of how it's structured. So often you can import an entire package. So one of the packages here is the one that we generated for sampling graphs. So these two algorithms would be our ones with this BiDAG package. The BNLearn package is quite a big one, also R-based, which involves a lot of greedy search and hill search. And also, it's another kind of search algorithm in this space of graphs. GobNib is an ILP thing, so it's an exact solver, but you have to restrict your search space first to make it run. This is one of these new continuous embedding algorithms putting here. This is the traditional PC algorithm from people in Zurich. Daniel knows them well, I guess. We had actually another version where if you change the order of independence tests, you can actually improve in terms of speed and performance. This is a PhD student of Juicy who just finished a couple of weeks ago. This is another score-based thing here. This is another package in Java here, which does, along with some sort of search, has a lot of independence testing type things, which also allow for some hidden variables in the background. And then this list goes on, but to give an idea of the variety of different algorithms that exist. And how it works on the inside is exactly through these JSON configuration files. So here, just some snippets. There's a lot more detail you can look up on the help online, but everything is interfaced through these JSON files. So for example, if I want to do a benchmarking thing, I just need to say, okay, I want to benchmark. Here's how I generate my data. These are the modules I use and the parameters I put into them. How I want to evaluate it. So here, if I want to do a ROC curve, what I need to put in for the plotting. How do I want to plot my stuff? And here, the main thing is what sets of algorithms I want to include. And for every algorithm I include, I then have this thing on the right-hand side. So I have the algorithm, I have idea, I guess, and I have some parameter I want to change for a ROC curve. So by default, it will go through all these values and then it will draw them on the ROC curve. And all the other parameters I can possibly put into this algorithm, I can define here. Or I can leave them as the default. So you have complete control over every algorithm and all its input parameters you can define inside this JSON file. And then everybody knows exactly which ones you use if you want to share the JSON file with other people. So you need this for every algorithm you want to use. Then you need to call a set of algorithms, and then you say, okay, what do I want to compare, etc., etc. And in terms of comparisons, maybe I'll go here first of all. So we have kind of built in, maybe remember from the purple boxes on the previous slide, we've built in support for these ROC type curves in terms of performance. You can check the runtime, of course. SHD is a measure of distance between two graphs based on how many distance but of structures. You can look at the matrices. I'll show up a lot later. You can look at the graphs themselves, and actually the nice thing is you can look at the difference in graphs. There's packages that allow you to do that too. And for these more Bayesian versions where we have a sample, we can look at the diagnostics of our chain. And so here I wanted to kind of focus on kind of a reasonable scale study. I do 100 repetitions, quite moderately large graphs with about 80 nodes. So it takes a bit more time, it's a bit more challenging to learn this side. We generate random graphs with this option in the PCAlg package. So developed people in Zurich. Here I'm going to look at two cases. So for Gaussian data, continuous, and for binary data. two different sample sizes. I think I only show the plots of one sample size to save time. And then inside we have one main parameter, the most important one, which we vary inside the JSON file. And this allows us to build these rock type curves. And of course, we save everything, the graphs, the parameters, time, and all the metrics of comparison. And here I want to kind of highlight one of the things you can do with this package. So once you've installed it, then you can run this thing very, very easily. Here, instead of doing all 50 algorithms, we picked a subset. Otherwise, you'd have 50 lines in the plot and it'd be a bit hard to talk about. So we picked a representative sample of here 12 in the continuous case and 11 in the discrete binary case. And I wanted to spend a bit of time to go through what this plot shows. So here on the y-axis is the true positives, rescaled by the number of positives, and the x is then the false positives. And here for each algorithm, we move through one parameter to build a rock type curve. So here at the bottom is the PC-alg. So this does independence testing of pairs of nodes conditioned on other sets of nodes. And this threshold here would be based on the significance level of my tests. So actually in the JSON file back here, it's this alpha here. Okay, actually, there's 4 values, not 3 values, but this is the thing we're changing. And you can see, I mean, it works, it learns graphs, but it's not particularly competitive compared to the other algorithms. The next one in the rock curve is this node here. So this is a continuous embedding. So it's very highly cited and popular algorithm, but if you do a fair comparison, it doesn't perform very well either in terms of performance or even in terms of time. The next set is this is the dual PC, so like PC, but changing the order of tests makes it faster and more efficient, does a bit better than PC. The these two here are from the BNLearn package, so simple search strategies in the structure space, which are moved by changing one edge at a time. So both here, hill climbing and a taboo type search, which is also hill climbing where I remove things I've already seen. Then this one here is the first hybrid algorithm from Smartanos about 10, 15 years ago. So it's like PC-alg, and then I do a score-based search inside, and you see it improves compared to that. And then the next set of performing, at least in this simulation setting, it depends, of course, on the size of the graph, the density, and other things. Now you can change all these parameters in the JSON file. Greedy equivalence search is actually quite an old algorithm, but it works in the space of equivalence class networks. And then here, this is an ILP, so an exact solver, but where we first have the pruner space to make it run in the first time and make it run at all. And then up here are 2 very recent search-based things that work on the space of topological orders. So if you go down the graph, you have an ordering of the variables. They form pretty well. These actually just from last year, these 2 algorithms. And actually here in the top are the algorithms that we developed. So using the Bayesian type approach to one search through the space, and then in purple, you probably can't see it. If we then take a sample and we use the posterior certainty of edges, we can actually filter out some false positives and improve our performance a little bit towards the left and top left-hand corner. And the nice thing is in this measures here, the SHD, so this Hamming distance in terms of structures, is actually just a Manhattan distance to the top left. So you can see already that SHD depends one on the parameter I pick and heavily on the algorithm I choose to. And so we quite like these rock curves because often you see people compare a set of algorithms for one choice of parameter. Because if I pick this one here with a bad parameter and this one with a good parameter, my relative performance can be influenced enormously. With here, the rock type curve, you at least can kind of compare a bit more fairly across choices of settings. Of course, picking the right setting in real life is much harder, but at least you get an idea of how well they could perform if you did know the right parameters. And if I go from the Gaussian case to the binary case, now if I imagine discrete data has a lot less information per observation than the continuous one, you can see the overall performance drops dramatically. So here we get very close to the perfect performance here. It's relatively small size data. Now it's only 320 observations for 80 size network. But we have a drop down to, only getting a half or at best 2/3 of the true edges in the graph and about 10% false positives. And you see a similar pattern here. So the PC-alg, actually the PC-alg is down here, number 10. These are score-based searches on the right-hand side here. So here they're adding a lot of false positives. And if I optimize for the score, especially for this binary data, I tend to find a lot of false positives. So here, this is an order-based search. These are the ones from before. This is the exact search. These are the hill climbing and taboo search. The ones that perform a bit better are the hybrid algorithm here. Our MCMC algorithms are 7 and 9 in this case, so sort of in the middle. And the ones that perform well again are the ones up here. This is the Boston graph, so new order-based searches. And also here, the greedy equivalence search actually works pretty well in this case. So you get an idea of, how well it can work. And yeah, if you wanted, you could run all 50 algorithms and have 50 lines and really check every single algorithm that exists out there. So when we were trying to publish this paper, one of the referees or some of them complained, oh, what about algorithm X, my favorite one? Or algorithm Y, how do you know it's better than this one? And so, of course, you do those comparisons in the revisions, but now we can really say, okay, if I compared every algorithm that exists, we can really check, where's it better, where's it worse, where do I see, good and bad performance? So if you ever want to make your own algorithm, this allows you to compare fairly, if you like, every single thing that really exists and has open source software. Another nice plot, at least for smaller networks, is to really compare the difference in graphs. So I can look at the graphs themselves, but you can actually check the difference. So here I can compare the estimated graph. So here we're looking in the pattern graph space, which is the equivalence class space. The correct bits are black, missing edges are blue, and red ones are incorrect. And you can see this part of the graph up here is not learned terribly well, but actually down here I'm doing pretty well. So you can already see on the previous plots, I get some distance based on the red and blue, but maybe I can see, okay, it's this part of the data that I'm really struggling with. Here's where I want to work more. And because we come from a more Bayesian idea, we run our chains, we get different samples. We want some idea of how well they're converging. So inside the package, we also have options for the usual trace plots. Check how my sample score is doing over time. And more importantly, if I have my chain, I have my sample of graphs, how much autocorrelation is there in my sample? So we know in MCMC we have autocorrelation, the sample's not independent across the chain. And here you get an idea, after maybe 30 steps, my autocorrelation is pretty low, so I can start to think, they're more independent. I get an idea of my effective sample size from these sort of plots. So if you're into MCMC, then these plots are quite nice for at least checking, if my algorithm's looking well, how much sample do I expect to have from my chain? And if not, how much longer do I need to make my chain to get good samples? And then finally, I wanted to show, so check the time, another plot where you can look at the graph, not as an actual graph, but as a adjacency matrix, like a heat map. So if it's a true graph, little blue dots where there's an edge, I can check, and particularly in terms of the Bayesian versions, I can have an idea of the uncertainty in each edge in terms of the posterior. I can check, do I match the edges, and where do I get uncertain edges? And where am I more certain about the edges being there in terms of my posterior sampler? And this is also part of the reason why we get better performance if I use the information in these edges. If I remove weak edges, actually improve my performance in the ROC curve with some thresholding. I'm getting close to time. So I hope these sort of figures give you an idea of what you can do if you're into structural learning causal discovery. This package that Felix develops gives you a lot of options to do these things. It's done, basically all the hard work has been done for you. So if you're interested, then the main thing is to install it locally for yourself. So here's 2 links to the GitHub, and then there's also documentation too about how it works. But the basic steps are, you go to GitHub, you clone, you download the Git repository, and then you have 2 main choices. Either you run it in a Conda environment, so you use like Aptena on Linux and the Conda environment, or you can go for a Docker container, so in like Linux, Mac, and Windows, it works in those cases in the Docker. And inside there, you just do your snake call, and you run the JSON file you've defined for your experiment, for your benchmarking. So once it's set up, installing it is very easy to run. As I say, either you can do it with Aptena, so only in Linux, or if you have other operating systems, you have to use this Docker solution. But all the documentation is there online. It's very well written, or hopefully easy to follow. And if you're into these tools, I guess it's very easy for people listening here to use these things. And the thing I really want to highlight before I finish is we added the optionality to add your own algorithm. So if you're in this field and you're developing new algorithms, you can easily install BenchPress, add your own algorithm, and we have like templates for doing so. So here, for example, if you're working in R, I can just take this template file here, replace it with my own algorithm, So here's an example on the right-hand side. I have my SnakeMake interface bits, no, to tell it the input and the output at the top and the beginning. And then the main bit you need to change is this lines here. So this is just basic code that generates a random matrix. No, so not based on data, no, it outputs a random matrix. So this bit you could replace with your own code, and then already it's in BenchPress, and you can run it against all the other algorithms already in there. The only other thing you need to do is change your JSON template, no? So you say, these are the parameters I put in, no? Make sure it matches up here, and then it knows in the JSON template, this gets forwarded for the algorithm used inside, and it comes back out. Yeah. So this hopefully makes sense. It's very easy to do. And of course, because it's all modular, it uses this whole SnakeMake workflows being built for us. You can add your own modules. You can even then push it to the repository and add it to the BenchPress as a whole. So these options are there. And we think they're very powerful. Like, instead of everybody running their own local simulation, you can use this stuff, or use SnakeMake and containers and all the work that Felix has done for you and get it set up. And once you set it up, it's very, very easy to do this. So I'd like to finish by one again highlighting the software and the link. If you're interested, please have a look. And it does allow you to benchmark this stuff on a massive scale that wasn't really do before, and do it reproducibly and much easier than before. Exactly using all the tools, from computational reproducibility that we've heard about also in previous things here. And exactly that, it's reproducible. You just share your JSON file and everything is in there essentially because it's all done in the containers. Many algorithms, so you can easily add your own. In terms of take home, in terms of you want to use one of these methods, then basically search and score offer the best ones. But you have to be careful which one you pick. Yeah? So there are a lot of search and score methods. These two work quite well, the BOSS and GRASP at the moment. Others can be quite poor. And we'd always advocate for search and sample, get an idea of uncertainty using these Bayesian inference schemes that we've developed. So this plot here, I kept one where we look pretty good now at the top left-hand corner. And with this, I'd like to obviously thank Felix for his hard work in making BenchPress, and Juicy, of course, at the University of Basel, and you all for your attention. I'm happy for questions. I think also Felix is here as well, maybe if you have questions for him.
Thank you very much, Jack, for this unusually theoretical talk we're having today. That's fantastic.